Okay, so we're talking about how the mRNA molecule that has just come from transcription out of the nucleus can code for this growing amino acid chain in our ribosome, and how tRNA molecules help with that. Okay, let's take a look, see what we got. So here's our ribosome and our mRNA molecule right here, and our transfer or tRNA molecule. So 3 nucleotides in this mRNA molecule is called a codon. And it's like 3 letters in a word. So these 3 letters make up the word valine, or it codes for a specific amino acid. And this word or codon, GUC, codes for the amino acid valine. And, get my pen here, this transfer RNA molecule can attach specifically onto a valine amino acid. So this right here is also a valine. Valine amino acid. And so this tRNA molecule floating around in this cytoplasm with the help of, guess who? Another enzyme. An enzyme will attach this valine onto a tRNA molecule, but a specific one. So this one right here that has the code CAG. So this transfer RNA will then be attracted to these 3 nucleotides on the mRNA, because cytosine is attracted to guanine, adenine is attracted to uracil, and guanine is attracted to cytosine. So this tRNA molecule will come in, dock in our ribosome, bringing with it our amino acid. And the ribosome will then facilitate the bond between the last amino acid and valine to make our growing protein chain. So this sequence of nucleotides, these 3 nucleotides or codon right here, those, let's see, I've got it written down, UCG, that codes for another one, amino acid called serine. So this amino acid right here is serine. This this codon right here codes for phenylalanine. So this amino acid right here is phenylalanine. And it's these tRNA molecules that are bringing in the amino acids according to their codons, or sequence of 3 nucleotides. Okay, so something that's kind of interesting to notice here. Here is our mRNA strand. Actually, let me get my eraser, and I'm going to erase, let's see, these. So the sequence right here, GGG, the complementary bases for GGG in our DNA, what was that? It was C, C, C. In mRNA, it's GGG, and in the tRNA, it's, again, because it's complementary, CCC. For UUU, what would be our, remember now this is DNA, so it would be A, A, A. And what's our tRNA? AAA. And for UCG, what was the DNA? Let's see, so UCG, it would be A, G, C. And there it is in the tRNA, AGC. And this one's interesting. Let's look at this one. So GUC in the mRNA, in the DNA, what would have been the complementary bases in DNA? They would have been C, A, G. And they're C, A, G in our tRNA. So these right here will be very interesting to take a look at. We can guess they're A's because of the color. So I'm going to say in our DNA molecule, in our mRNA, they're A, 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 A. What were they in the DNA? So remember, in DNA, adenine bonds with thymine. So it's going to be T, T, T in our DNA molecule. And then in our mRNA, it's going to be AAA. So what would it be in our, let's see if I can draw this. tRNA molecule with our amino acid on the tip. So remember, there's no thymine in RNA. So it's going to have to be U, U, U. So UUU is going to come in here, bringing in its specific amino acid to be attached to this chain. So that's how this code from the DNA gets transcribed into an mRNA, and then the mRNA docks with a ribosome, and these tRNAs then bring in these amino acids specifically according to the nucleotide sequence in our mRNA. Okay, so why then, why three? Why 3 letters? Well, let's think about this. Okay. Let me get my pen. We've got think I'm going to put it up across the top here. So we've got 4 nucleotides. Actually, I've got some notes I need to look at here. Okay, there we go. So I get it all in the right order. So we've got uracil, cytosine, adenine, and guanine. And there are 20 amino acids that are needed to build the proteins in our bodies. Can we build 20 amino acids with 4 nucleotides? If we code for 1 amino acid for each nucleotide, we can then come up with only 4 amino acids. So that's not going to work. What if we combine the nucleotides, say, into 2-letter words? Would that be enough to be able to build 20 amino acids? Well, let's try it. Okay, let's say our first letter is, let's say, oops, no, don't want to put it over here. So U, C, A, G. Oops, sorry. Same order. So our first letter, U, C, A, G, and then our second letter is U, C, A, G. So U, U. Let's say that UU codes for an amino acid. Let's say that UC codes for an amino acid, and UA codes for an amino acid, and UG codes for an amino acid. And we keep go all the way through. How many amino acids can we get? We can only get 4 times 4. We can only get 16 amino acids, but we need 20 amino acids. So that's not enough. So what if we then have 3 letters to code for an amino acid? And that works. Is we have the first letter, UCA or G, we've got the second letter, UCA or G, and then the third letter, UCA or G. So how many amino acids can you then get with 3 letters? And you can get 4 times 16. So 4 times 16, or 64. And that's plenty, with more to spare. So this chart right here is codons of mRNA and which amino acids are attached to our protein chain if the mRNA has this codon right here. The amino acid would be phenylalanine. And then these combinations right here will be all of these other amino acids. So there's actually 61 codons that code for amino acids. 3 of them code for stop. So it tells not only